DNA replication as the process by which a cell copies its DNA before cell division. It's described as semi-conservative, which means that each DNA molecule consists of one original strand and one newly synthesized strand. So this diagram here represents the process of DNA replication. Now, the first step, it begins when the enzyme helicase unwinds that double helix. Alright, so where's helicase in the diagram? This one here. So this little wedge-shaped thing, that is DNA helicase, and it unwinds this strand here. So it unwinds the strand by breaking those hydrogen bonds that are between those base pairs. Now it separates it into two strands, which forms a replication fork. So that's what it says here. Replication fork is this point. Okay, the point where it's separating those base into two separate strands. Alright, the next step, DNA polymerase adds the new nucleotides. So where is it? Here's DNA polymerase, this big yellow thing. It adds new nucleotides to each original strand. Okay, in a 5' to 3' direction using complementary base pairing. Okay, so this is the leading strand here. Now the way in which DNA polymerase adds is in a 5' to 3' direction, which just means that this end will be the 5', and then this end over here will be other way around. So this would be 3' of the leading strand, and this would be 5' of the leading strand, of the original strand, but it's adding the bases, so it's actually putting them in a 5' to 3' direction. Alright, so this way it's going from the 5' to 3'. Now, this is happening on both the original this way, and if it's adding in this direction, that means that it's the leading strand. But on the lagging strand, DNA is synthesized continuously in the direction of the replication fork. So that's this one here. So it's adding in this direction, which is towards the replication fork here. That's in the leading strand. On the lagging strand, DNA is synthesized discontinuously in short segments called Okazaki fragments, because it's running in the opposite direction. It's running away from this replication fork, which means that it's just to add in fragments, and these fragments are called Okazaki fragments, and they're running in the opposite direction. So these Okazaki fragments, so this would be an example of an Okazaki fragment, this part here, they're later joined by enzymes, okay, to form a continuous strand. So this one is the leading strand, and it's working towards the replication fork. So it's adding these bases, complementary bases, and it's running continuously this way towards the replication fork. But the other one is running in the opposite direction, backwards this way. It still adds in a 5' to 3' direction, but it's working in an opposite direction, which means that it's going to have to make it in chunks, and those chunks, or those fragments, are called Okazaki fragments. Okay, so to summarize, DNA replication involves helicase unzipping the DNA, and then DNA polymerase builds new strands, and then the Okazaki fragments on the lagging strand, this one here, will complete the process. Okay, so that is Very quick summary of DNA replication.